right, to overcome the blood feeding. And certainly I'm not the first person to think of this. And the picture that's going to pop up in a minute is Professor José Ribeiro. Oops. I thought he was. There he is. Who is really kind of the father of this field, characterizing different molecules in the saliva of different blood-feeding arthropods. So the idea here is that we could take advantage of this fact to try to stop transmission. So if you think of what I like to call the unholy trinity, we have pathogens, hosts, and vectors. There's really different areas in this triangle that you can block to stop transmission, right? You can have vaccines, where you vaccinate the hosts against the pathogens to stop the transmission cycle. You can use insecticides or other barrier kinds of methods to stop these arthropods from biting the hosts. You can make transmission blocking vaccines to block the pathogens infecting the insects, or other kinds of control that I think you may have heard of earlier in the course, of ways to block this from coming. So here we're actually talking about saliva or spit of these insects in a way to influence the immunity of these hosts to these pathogens. So as I said, these blood-feeding arthropods, they inject these molecules because they need to blood feed to make their eggs. And when they release these salivary proteins, they're immunogenic. That is, they activate the immune system. So you've all probably been bitten by mosquitoes and you have this swelling. And here's the arm of my graduate student. And you can see the redness and the swelling, and that's the immune response reacting to the saliva that's released into their body. This is actually the arm of an individual who's been bitten by sandflies, and that reaction is quite drastic compared to that of mosquitoes. So there's this idea in the field of parasitology. It's not working. Good, thank you. Very good. Can you hear me now? Yeah? Okay. Sorry. Thanks. All right, I don't know where did I end? So immune response from spit, right? You have an immune response to spit. So the other thing that's an idea in parasitology, it's certainly true in some cases, that people who live in endemic regions are less likely to get disease. We know that's for sure for malaria. And in some places, we think that's also the case for leishmaniasis. So if they get the disease, it's attenuated, so less susceptible. The other important fact are that flies or mosquitoes, they're not in a population are not very infected. So even in the most highest transmission places of malaria, for example, only 10% of those mosquitoes are infected. And for sandflies, it's much, much less for leishmaniasis. So even though you may only see infected sandfly every now and then, you may be getting bitten by 100, 1,000 times a day by a sandfly or a mosquito, whatever the case may be. And each time it's injecting saliva, and each time you're getting an immune response. And so you might imagine a new individual, a visitor, a traveler, a tourist comes to an endemic site, And it's the first time they're exposed to that saliva, and they happen to get bitten by an infected fly. That microenvironment in that individual would be different than someone who is being bitten all the time and has a different immune response. And so that's this idea, is that we could mimic that and vaccinate people who aren't exposed to change that reaction. When this idea first came out, it was like, let's make a saliva vaccine for all blood-feeding arthropods, for all insects. But as you can see right here, the response to mosquito bite is different than a response to a sandfly bite. And in fact, the response to different mosquitoes and different sandflies tend to be different. So there's a lot of vector diversity. And actually, that's one of the main things that José Ribeiro has found, is that there's a huge diversity of salivary proteins in mosquitoes. So it may not be that we'll be able to make a vaccine using a saliva that targets all blood-feeding arthropods. Oh. So actually, this is a table from a review from José Ribeiro. And one of the things you can see is, if you look at the total proteins or total families found in both culisines and anophelines, there's 83 different proteins, but there's groups that are unique to each individual mosquito. So even though blood-feeding arthropods have to overcome the same mechanisms and they have the same functional activity in much of their saliva, that functional activity is performed by different proteins and different molecules. So quite a bit of vector diversity. So when you think of a vector-transmitted disease, we have all this vector diversity. Certainly we have mosquitoes, we have ticks, we have black flies, we have sandflies, lots of diversity, lots of taxa that transmit pathogens. But even within one disease, there's quite a bit of diversity. So leishmaniasis, or really leishmaniases, as you all know, is a group of diseases, and you have different clinical outcomes, from self-healing cutaneous lesions to visceral disease that can be fatal, to many other clinical spectrum in between, often dictated by the species of Leishmania that initiates the infection. So we also have a lot of pathogen diversity in that aspect. Oh, sorry, I'm not used to your clicker here. So one of the things that we know is that we have the pathogen diversity, but there's a close evolutionary relationship between the pathogen and the vector species, right? So that in nature, often a vector or certain species will only transmit one or two parasite species. And so you can see this here for phlebotomus, which transmits Leishmania parasites in the Old World. And we have different Leishmania species, L. major, L. tropica, Ethiopica, Donovani. And there's only a few vectors that are competent to transmit those particular species. And so that actually, there's some diversity there. But it's nothing compared to the New World, as you might know. So these are all the different species of leishmaniasis. Yeah, I know you can't see them, but there's lots of different ones and lots of different vectors. So there's a huge diversity of trying to target to make a vaccine. Whether you're trying to make a vaccine just to the parasite itself, but if you're trying to incorporate spit or insect saliva in that vaccine. So studying leishmaniasis, we often use a model. And in fact, immunologists use leishmaniasis as a model to study immunology. They don't even care about leishmania. And this model is in inbred strains of mice. And in general, the model is injecting the infectious parasite into these mice without the sandfly. So without even caring about what the sandfly is doing, because we can culture these parasites in the lab without the sandfly. And so essentially what the model is, most often it's Leishmania major, which is an old world parasite, but we also study this with in mice with visceral parasites, as well as Leishmania amazonensis, brasiliensis, mexicana, and it's subtly different depending on the pathogen, but generally the experimental model is the same, where you inject the parasites either into the foot pad, or here's the ear, and this is a cutaneous causing parasite, and you have the lesion, and so essentially you just measure the lesion diameter or the thickness, and you can plot that on the y-axis, and in general that's equivalent to parasite number. Not always, but you can say that's how the disease is progressing over time. And we have resistant mice that are actually recapitulate what happens in humans. So in most humans, in response to this pathogen, you get a lesion, you eventually heal that lesion, and you become immune for life. So on the opposite end of the spectrum, we have a susceptible mouse, which never heals and just keeps going and going until essentially the ear would fall off if you let the experiment go on. And so immunologists have actually utilized this model to study Leishmania, but also to characterize the immune response that leads to this resistance. So now you can also, even though immunologists have done this, you can also do this transmission with a sandfly, actual natural transmission, and you recapitulate essentially this same difference between susceptibility and resistance, except there's a little caveat because it's injecting saliva when it does a natural infection. And that changes a little bit, and we'll talk about that in a moment. So researchers, David Sachs' laboratory at the NIH in the US, posited that this exposure or pre-exposure to these uninfected bites that people living in an endemic region which occurs could influence the immunity to the Leishmania. And so they used this model that I just told you about, where they inject parasites into the mouse ear and they measure this lesion. And then they tried to mimic what it would be for a visitor or a tourist coming. And they actually isolated saliva from sandflies, Phlebotomus papatazi, which is a natural vector for Leishmania major, and they injected the parasite in the presence of saliva. And what you can see is this exacerbation or enhancement of the lesion. So indicating that the saliva is increasing that particular response. So then they wanted to mimic a person that was living in the area, getting bitten lots by uninfected flies. Okay. So then they vaccinated or mimicked an uninfected fly bite by vaccinating with saliva alone 3 times, after which they challenged the parasite in the presence of saliva. And what you can see is certainly the lesion size is reduced from this enhancement, but what was interesting is it's even enhanced from the pathogen alone, suggesting that whatever that immune response is to this treatment, it's creating an inhospitable environment. So the group, Schott and Kamhawi, actually repeated this with natural exposure. So instead of needle and saliva, she actually took uninfected sandfly bites and treated the mice with uninfected bites, and then she challenged with an infected bite. And so in mice that were not treated ahead of time or bitten ahead of time, they had a lesion that healed, but if they pre-exposed them to uninfected bites, you see this huge resistance response. So this set off this whole field of research of trying to use saliva in a vaccine component against leishmaniasis to try to help create a vaccine, because we don't have a vaccine. But the question that we need to think about is, when we make a vaccine, are we just getting rid of the enhancement? Or is there some sort of bystander effect that influences immunity directly to the parasite and not to the saliva in this context? And you can imagine that you could do either. So this is done in inbred strains of mice with flies in colony in the laboratory. And studies started coming out suggesting that maybe it doesn't always work. You know, these mice that we were working in are pretty they are homogeneous, they're inbred. And so that maybe this wasn't a real phenomenon, just an artifact of the laboratory. So I had a graduate student who did a meta-analysis. So she took every figure from every paper that she could find that looked at saliva and a vector-transmitted disease. Turns out the vast majority of those papers are with leishmaniasis, various species of sandfly saliva, various species of leishmania, and very few looking at others. And so she did this meta-analysis to see if, in fact, there seemed to be a global difference to saliva. And so this is just, if you're to the right of that dotted line, it means that you have a response. So what she discovered was that it seems, if you look at all of these different experiments, saliva certainly causes an enhancement if it's in a naive individual, like a tourist, and so that holds up statistically. And pre-exposure to mostly leishmania, not so many other pathogens, there wasn't enough power in the statistics to say yes or no, leads to a reduction in pathology or a reduction in the lesion size. When she tried to ask questions about the type of immune response that was activated, either by one exposure to saliva or multiple exposures, there wasn't enough evidence to say either way. And certainly she asked the question, well, what happens with humans? Does this work in humans, which is probably more relevant? And again, there were not enough studies to say at that time, and I don't think there are either now. So in the next few minutes in this first session before our break, I'm going to go over many of these studies with these different parasite species and sandfly saliva, because it illustrates that everything is not exactly the same. And so there's some differences that if we're going to use it as a vaccine, we need to think about. So Jesus Valenzuela was a student of Jose Ribeiro's, and he's carried on this work, really characterizing the spitome of all of these vector species, some mosquito species, but primarily sandfly species. And so essentially what he does is he takes the salivary glands of a sandfly, mushes it up, runs it out on a gel, and then cuts out each of these proteins and sends it to mass spec to find out what it is. And so what you can see here is there's quite a, by the number, there's quite a bit of diversity in these sandfly species. There's some common proteins or homologous proteins, but there are many that are not the same. And so Jesus and the others wanted to find out in this Phlebotomus papatazi model, which I showed you before with the salivary glands that leads to resistance, they wanted to find out what proteins were mediating that immune response to lead to this resistance. And so they tested that. So they vaccinated with different proteins. At that particular time, they didn't have recombinant proteins, and so they cloned the gene. I mean, there were recombinant, that technology was available, but they had yet to express those recombinant proteins. So what they did was they cloned the gene for each of those proteins and put it in a plasmid, and then they vaccinated with DNA. right? So the idea is you vaccinate with the plasmid, then the host actually expresses that gene, translates that gene into a protein, and then it's a vaccine. When they vaccinated with control DNA, so the plasmid alone, they saw a lesion size that you would expect. When they pre-treated the mice, this was without vaccination, this was just by sandfly bite, exactly what I showed you before, they saw resistance. So what we would expect. When they vaccinated with salivary gland homogenate, so when, like I showed you before, they took those salivary glands, mushed them up, and injected them, they also saw resistance. Now remember, that's a cocktail of all of the proteins in both of those cases. They found one plasmid expressing one gene for salivary protein 15 that recapitulated that response. When they did the others, oh, there it is. One, for example, SP42 that's on this graph, it did nothing. So it didn't seem to be inducing immunity. Most importantly, another one, maybe, is it going to come up? Oh, SP44 actually caused an enhancement. They had to stop the experiment at this point because the lesion size got so big. And so this is important because in this cocktail, immunity to SP15 seemed to overcome the immunity to SP44. But in general, if you're going to make a vaccine, you're not going to mush up salivary gland homogenates and treat lots of people. You're going to try to express them recombinantly. And so you're probably not going to express all 30 proteins in a vaccine. So choosing the ones appropriately are important, because if you accidentally had too much of this, you might get the opposite effect that you want. So protein diversity. We have vector taxa diversity, we have parasite diversity, we have protein diversity. All of these things that you have to think about. So the other part to vaccine development is trying to find out the proteins that are responsible for immunity. Is you have to create a system where you can do this fairly quickly. So cloning out every gene that every blood-feeding arthropod has to make a salivary protein, and then expressing it recombinantly, or doing DNA vaccine or vaccination on mice, and then challenging. All of these things cost a lot of money and take a lot of time. And so often we try to have a surrogate marker, if you will, to look for that immunity so you can do it quickly. And so Jesus's group wanted to find out what kind of immunity was this response so that they could eventually use that as a surrogate to test. So they did this experiment where they wanted to see if cellular immunity or humoral immunity was important for the resistance response. So cellular, usually we think cells. Humoral, we think antibodies. And most vaccines, if not all, work by inducing antibodies. And so they went to test that. And so the same model that I showed you before, but they tested in B cell deficient mice. B cells are the cells that produce antibodies. So in these mice, they can't make antibodies. And when they vaccinated with SP15, they still saw that resistance phenotype, indicating that antibodies are not necessary for this response, and that it's a cellular immune response, or, as we'll show you in a little, a delayed type hypersensitivity, which is a cellular response. So when you're making a vaccine, you always have to think about humoral versus cellular. In this particular case, Leishmania is an intracellular pathogen, and so cellular immunity is appropriate for causing resistance. So they were looking for this DTH, and they wanted to look on different hosts and using different phlebotomous species to find the protein molecules that cause this delayed type hypersensitivity response. So when they did the mouse, and this is what I showed you before, SP15 caused a DTH, which is really a swelling. It's this response that you see when a person gets bitten by a sandfly, this hard red dot. And it certainly caused a DTH and caused resistance. If you remember, SP44 didn't really do anything. SP42 caused a DTH, but it made the lesion go further, right? So you have to be careful on what you choose. When they change the host, however, they have some that are the same, but many of the molecules are different that cause this DTH response. So now we have diversity of the vector, diversity of the proteins, diversity of the pathogens, and host diversity. So depending upon where you're trying to vaccinate, you might have to change what your protein is. And you have to know that going forward. So if it's humans, you know, what's happening in humans, and you need to be careful of what you choose. Most recently, this group tested this resistance phenotype in Rhesus monkeys. So normally when you're making a vaccine, you start in animal models, or you start in a natural system to see if immunity can even happen. Then you start in an animal model, like a mouse model. Then you move to a non-human primate model, and et cetera, and you keep going. So in this particular case, they tested it in Rhesus monkeys. Oh, I need to fix something. Just go to, here, just go here. We're just gonna move that off. I don't know if something's wrong with the animation. Okay, make it go down, and then just make it go. Actually, put it back. Yeah. No, here. No, let me do it. Sorry. There we go. So they tested this first using bites. So they had monkeys that were not exposed and those that had been pre-exposed. And in this case, they used Phlebotomus dubosci, which is also a natural vector of Leishmania major. So why did they switch their model? It's always good to test other vectors to make sure that it's working. But in reality, which happens in the lab a lot, things just stop working. So they switched from Phlebotomus papatazzi to Phlebotomus dubosci because somewhere in these studies, their sandflies stopped transmitting Leishmania. They just stopped transmitting, and they couldn't really, they'd spent many years trying to figure out why and haven't been really able to sort that out. So for students, this is a good lesson because things just biology happens and we don't always understand what's happening. Nonetheless, they were able to recapitulate in mice, which I didn't show you, the same thing that I showed you before, but with Phlebotomus dubosci instead of Phlebotomus papatazzi. And they're able to do the same thing here in the mice. So those mice that were pre-exposed by uninfected bites have reduced disease burden, which would be just lesion size. They also did another measurement, which you can always do different kinds of measurements when you're making a study, where they just took the maximum lesion size, and that's also reduced. And if you remember the picture of those lesions that I showed you, sometimes there was just a little bump or induration, and sometimes there was a big icky ulcer. So they all, and when it becomes ulcerated, often it scars and leaves a nasty scar. And so the other measurement they looked at was the percent that actually had ulcers. And the naive mice, more of them, those lesions actually ulcerated than the ones that were pre-exposed. So this vaccination with uninfected phlebotomist Dubosky bites seems to work in response to Leishmania major. One thing I could mention here, which is important, is when you read a title of a paper, which a lot of people just read the title. They don't even read the abstract. But even, it's a trick that scientists do. We write a sexy title, and then we write in the abstract, our message. Not always does the data in the paper really support what we're saying in the title and the abstract. And so sometimes you'll see, saliva as a vaccine protects against Leishmania. And if you saw that, you would think, yeah, it's a vaccine. We're gonna save the world of leishmaniasis. But it's not a sterile vaccine, right? You still get lesions. You still get ulcers. It's just modifying the response. So it's always really important as scientists to read the paper and realize what actually is being done, and not just go with the headline that you might see on CNN or I don't know if you have similar Fox News. I think you have a similar one here to Fox, yes. So it's important to actually know what they did and how they did it. All right, what's next? Whoops. Oh, sorry. Okay, stop there. All right. So then they wanted to see if this SP15 vaccinated, if it worked, right? And so they did a similar thing where they vaccinate with DNA. They vaccinate twice, and then they actually boost with recombinant SP15. And so vaccine research, when you actually get to the part of how you're going to deliver the vaccine, I personally think is a little boring, because you have to just do experiment after experiment with time. When should I vaccinate? Every 2 weeks or every 6 months? Should I vaccinate with DNA? How much DNA? With protein? All of these kinds of things. But they've done all of this and developed a good vaccine regime. And so they vaccinated with SP15. Now it's phlebotomist Dubosky salivary protein 15. And this is not in the presence of challenge. This is just looking to see if they can get their DTH response in the mice. And so induration size is just that swelling that happens with a delayed type hypersensitivity response. And they saw that they did get quite a bit of response, but there are some monkeys that don't seem to respond to the vaccine. They went further and measured the cytokine, or interferon gamma, which is the cytokine or the protein, that host protein that's important for Th1 or a cellular immune response. And they see that some of the monkeys actually induce this response, but some don't. So we went from this inbred strain of mice where they all basically did it, and so now we're in these human primates, and we're getting a little bit of a variable response. Okay, you can play it and go to the next one, and then we'll be fine, I think. Go, play. Yeah, good, okay. Right, okay, good. So then they tried to test this, actually, to see about antibody response. So in general, you can think of a cellular immune response as the opposite of the antibody response in this particular case. So humoral versus cellular. And so they tested to see what was happening with the antibodies, pre and post-vaccination. So when they looked at the monkeys that produced interferon gamma from the previous slide, they saw a little bit of interferon gamma, but not very much. When they looked at the monkeys that didn't produce interferon gamma, they saw high antibody content. So these these monkeys have different responses. Then they tested it with challenge, and they found that the monkeys that had these high antibodies and no interferon gamma were not resistant. But the monkeys that actually had the interferon gamma response and low antibody were resistant. So it seems like the vaccine is working in these non-human primates, but maybe not as effective as we would want. Because when you go to a clinical trial, you just test lots and lots of different people, and you want a better efficacy than 30%, for example. So in this case, I guess 30% aren't working. And so as you go forward, you have to think about it's not going to be 100% yes or no. All right. So that was mostly talking about a natural model of transmission in the old world, Leishmania major and phlebotomus. And those studies that I showed you were performed over 15, 20 years. So it wasn't like they just did it step by step. I'm making the story a little bit quicker. In the meantime, there was lots and lots of other studies that were done and that continue to go on with different species of sandflies and different parasite systems. One of the first was actually by Richard Titus's group, and they were actually looking at Lutzamia longipalpus with Leishmania major transmission. Does that seem kind of weird? Okay, it's kind of weird. Why do you think that might be strange? Have you guys studied leishmaniasis at all? Do you guys know Lutzamia longipalpus? Lutzamia longipalpus is present in Brazil. It's a New World sandfly. Leishmania major is only found in the Old World. This is not a natural transmission pair. This will not happen in nature. So with sandflies, we have this idea of permissive and restrictive vectors. Have you heard of this? Okay. So in nature, I said there was the close relationship between a sandfly and its parasite, where you usually only see transmission of a certain parasite by a certain species of sandfly. In the lab, however, we have some sandflies, Lutzamia longipalpus is one of these, that is permissive, that if we take it into the laboratory, we can make it transmit anything. I mean, basically any Leishmania species. We also have restrictive vectors, Phlebotomus papatazi, Phlebotomus dabaski. They are both restrictive, that even in the laboratory, they are not competent to transmit anything but Leishmania major. So if you try to put Leishmania infantum, it will not transmit it. Nonetheless, this is a model that many people utilize. I'm not sure why, but they do. This is actually quite an old paper here, and they are trying to study the effect of sandfly saliva. And they inject Leishmania major in the mice, and they see this lesion size. When they inject Leishmania major in the presence of Lutzamia longipalpus salivary gland homogenate, they see an exacerbation or an enhancement. So this is interesting. We see that same enhancement with Phlebotomus papatazi saliva. So the two species of sandflies, which are probably, 100,000 years apart evolutionarily? I mean, that's like humans and whales. That distance is really, really, really quite large. You still see this exacerbation, which is interesting. Then they did the same experiment using a small peptide from the saliva, maxidilin, or maxidilin, you can say tomatoes, however, and they recapitulated that exacerbation response with this small protein, which actually is a really potent vasodilator. And so that's interesting. You can't see it, I guess, where you are, but this is quite a large amount. So antigen quantity is also important. When we're doing these experiments, for example, we're isolating the salivary glands of sandflies, and although they're much, much, much smaller than a mosquito salivary gland, there's still a lot more saliva, plus membrane components and various things that you're injecting into the mouse compared to what a sandfly would be doing, right? It's only releasing a little bit of those molecules. We also know that some of these proteins are activated in the salivation process. So when you isolate the salivary glands and inject them, they might not be the active component that you would see in nature. And then when you're vaccinating with this large amount, you may be inducing a response that wouldn't be natural. But maybe it doesn't matter if it works, right? So then they wanted to test if they could vaccinate, right? So here's what I showed you before. L major in the presence of Lutzomyelongiopelvis salivary gland homogenate. Here, they vaccinated with maxidillin, and then they challenged, and they were able to get rid of this exacerbation response, as well as that bystander effect. And then this is just infection with Leishmania major. So the question here is nature versus nurture. So even though what I just told you about injecting all those salivary glands or too much antigen is not a natural process, maybe that's what's not naturally happening, and scientists argue about that all the time, and we could argue about that all day. But maybe it doesn't matter. If you're trying to make a vaccine or a treatment, you can, even though it's not naturally occurring, but you can influence that natural response, maybe it doesn't matter. Scientists will argue both ways. So we talked now about SP15, we talked about SP42, SP44, and then we switched species to Lutzomyelongiopelvis. And we said one of these proteins that could be working in this response is maxidillin. Here, they test again Lutzomyelongiopelvis with Leishmania major, but they look at a different protein. Whoa. So in this particular case, they do a similar thing that they did with the phlebotomist, where they're vaccinating with DNA vaccination and then challenging. And so here you see empty vector, which is the response, the control response. If they vaccinate with Lutzomyelongiopelvis salivary gland homogenate, they get resistance. Several of the plasmids with different proteins don't do anything. One of them, LJM11, recapitulates what we saw with SP, Papatazzi, and Dubosky 15. So now we have different species, so it's not surprising that we might have a different protein. And remember, this is lesion size. They also tested that with recombinant protein and looked at parasite number, and they see that the control, some other proteins, you get this high amount of parasite number, but salivary gland homogenate and LJM11 gives you reduced parasite numbers. This recombinant protein also gives you a high amount of interferon gamma, or that cellular response. So they kind of recapitulate what we saw, but now we have a different system. All these responses, and the challenge in this case, are with Leishmania major plus salivary gland homogenate from Lutzomyelongiopelvis. So in this context, while we're showing it works the same with Phlebotomus papatazi, in nature, you will never get infected with Leishmania major in the presence of Lutsamyelongipelvis salivary glands, because that's not a natural vector host pair. So while it works great, I think you wouldn't want to make a vaccine with this protein for vaccinating against Leishmania major, because it wouldn't work, because you would never stimulate that immunity naturally. So again, natural versus unnatural pathogen pairs. So they wanted to test the same idea with visceral infection. So in this case, they used Lutsamyelongipelvis and Leishmania infantum chagasi, which causes visceral disease and is a natural pathogen vector pair. So this makes a lot more sense. And this is in hamsters, because mice are not a good model to cause visceral disease. So now we've switched host species again. And they did the same thing, and they looked for parasite burden in the liver and spleen, which was where visceral infection occurs, 2 months after vaccination and 5 months after vaccination. And you can see the control DNA and these vaccination protocols with these molecules do not give you any control. There's LJM11 that we saw before, and LJM19, which seems to be giving you resistance. But after 5 months after infection, LJM11 is not working in the way it did with Leishmania major, but LJM19 seems to work. If they look for survival, here's the control. Vaccination with LJM19 saves all of those hamsters from dying. And this is the antibody titer, so we won't need to worry about that right now. So the point here is we've changed. What have we changed? We changed the parasite species from Leishmania major to Leishmania infantum chagasi. We changed the host from mouse to hamster. And so just in those changes, we've had to find a different molecule or a different protein that led to that resistance. We also changed the timing a bit, right? So it seems like LJM11 worked here early. It just didn't give a lasting immune response. So the timing is also important to think about. So pathogen diversity. Let's try another natural relationship. So now we've switched to Lutsamia intermedia, which is also present in the New World, and Leishmania brasiliensis. And so this is a cutaneous infection in the mouse. Brasiliensis also causes mucocutaneous in humans, but we don't have a model system to study that. So same thing that we've been doing, vaccinating with salivary gland homogenate, and then challenging with parasite and homogenate. I'll finish this and we'll have our break. And so in naive mice, challenging with the saliva and brasiliensis, you get this response. If you pre-expose them to saliva from uninfected Lutsamia intermedia, you see exacerbation, right? For everything else, we saw resistance. So now this is a different pathogen species or sandfly pair, and we get the opposite effect. So that's an interesting, it may not work for everything. So you get an adverse response, not what you were expecting. So they set out to test, well, maybe that's true, but maybe we can facilitate a different response. And so we know that vaccinating with Lutsamia longipelvis salivary gland homogenates can lead to resistance. So maybe we can vaccinate with longipelvis and then challenge with Braziliensis with saliva from intermedia and change that response. So what they did, oh my gosh, is they vaccinated with Lutsamyelongipalpus, salivary gland homogenate, in one ear, and they challenged with salivary gland homogenate from Leishmania intermedia, right? And to see if they could cause resistance. So here's the control. They wanted to see if they could, so here's the negative. This is susceptibility. It works if you challenge with Longipalpus, but it also works if you challenge with Leishmania intermedia. Meaning that even though we pre-exposed with bites from intermedia, with bites from Longipalpus, we could cause a resistance response with the challenge with intermedia. What they didn't do here, which would be more likely happening in nature, is pre-exposing with both Lutsamyelongipalpus, or the vaccine, and intermedia saliva to see what the response is. But we're making steps in the right direction to try to incorporate the kind of diversity that we might see in nature. All right, then when we come back, we're going to switch a little bit more of what's happening in humans. So let's have our break. And I'll take any questions if you want to have any questions, but I heard you guys don't ask questions. You can come and ask me in person if you want.
Questions? Okay, I have a couple ones.
Of course.
Maybe it's a naive question. Sorry if it's a stupid one, but anyway.
No question is stupid.
Okay, but if we know the kind of cellular response which protects against leishmania, this cannot be manipulated using known drugs or known antigens which has no relation with leishmania. And
so he's asking the question about if we know what causes resistance to leishmania, and we do, can't we manipulate that in some way? So I will say there are some studies that suggest that the current treatments now, so the gold standard treatment is antimony treatment. It's horrible, right? But it suggests that that's actually inducing this kind of response. So in that way. We know in these mouse systems, so the susceptible mice that go way up here, you can treat them with recombinant cytokine. The timing is important. If you wait too late, it doesn't work. But if you do it early, you can make that become a resistant mouse. So we can manipulate it. The problem with type 1, Th1 cellular immune responses is we don't really know how to vaccinate to induce that very well. So like I said, almost all vaccines are antibody mediated. Basically, like, so for, how many of you had your influenza vaccine? Flu? You should go get your flu vaccine. They work by antibodies, so stopping the virus from going in. As immunologists, we don't really know how to activate in a vaccine component cellular immunity well that lasts a long time. That's also why this is an interesting phenomenon, because maybe we could induce that by tricking, because you're always going to be getting boosted by sandfly bites. So yes, you could, but we don't know how to do it.
Another question is about this idea of the cocktail.
What? Say again?
The idea of the cocktail of proteins which is present in saliva. Is it possible to do cellular culture using salivary gland cells?
Oh, and to like make it.
Yeah, like an hybridoma of salivary gland cells and try to produce. Because I was wondering, in the malaria, the best vaccine is produced. The best protection is produced with saliva, with natural.
I'm gonna talk about that at the end.
Okay. Okay.
Yeah. I probably won't get it a huge talk about it, but at the end I'm going to talk about that. I think it's important,
Okay.
for sure.
So thank you very much, Mary.
Thank you. Maybe we can have coffee or something to drink. Thank you.